Now coming towards immunoglobulin IgM. What's really interesting about IgM antibodies is that they are present as a monomer on the surface of virtually all B cells, where it functions as an antigen binding receptor. But in serum, it is present as a pentamer composed of 5 H2L2 units plus one molecule of J chain. The IgM is present on B cells and are the main immunoglobulins produced early in the primary response. Because of the large size of the pentamer with 10 antigen binding sites, it possesses great binding avidity and therefore is the most efficient immunoglobulin. In agglutination, referring to the clumping of cells, it is also efficient in complement fixation or activation and other antibody reactions, and is important in defense against bacteria and viruses that are associated with cold autoimmune hemolytic anemia. Cold autoimmune hemolytic anemia is an acute anemia in which primarily IgM together with the complement causes red blood cell agglutination and also extravascular hemolysis upon exposure to cold temperatures. Now, this results in painful blue fingers and toes. It is also seen in conditions like chronic lymphoid leukemia, mycoplasma pneumonia infections, and infectious mononucleosis. Remember that IgM does not opsonize directly because there are no receptors on the phagocyte surface. However, IgM activates complement, and the resulting C3b can then opsonize because there are binding sites for C3b on the surfaces of phagocytes. Now, IgM is involved in many areas regarding the body, but two syndromes that come up frequently are hyper-IgM syndrome and the Viscott-Aldrich syndrome. Hyper-IgM syndrome is a syndrome in which severe recurrent pyogenic bacterial infections resembling those seen in X-linked hypogammaglobulinemia begin early in life. Patients usually have a high concentration of IgM, but very little IgG, IgA, and IgE. And unlike the X-linked hypogammaglobulinemia patients, these patients have normal numbers of B cells. But instead, in hyper-IgM syndrome, the deficiency is in the gene encoding the CD40 ligand, which is also termed as CD40L, which CD4 positive T cells express to bind and activate other cells through the CD40. Now, the CD40L is one of the main components of T cells, and the failure to properly interact with B cell actually results in the inability of the B cell to switch from one production of IgM to the other classes of the antibodies. This too can be treated with pooled gamma globulins. Now, the Viscott-Aldrich syndrome, in which there are recurrent pyogenic infections, eczema, and low platelets, which typically appear during the first year of life. This is an X-linked disease caused by the mutation in the Viscott-Aldrich syndrome protein gene, leading to a defect in actin filament assembly that is important for the T cells to respond to the antigen presentation, and for B cells to be activated by the signals from the B cell receptor. These patients are therefore unable to mount an IgM response to the capsular polysaccharides of the bacteria, such as pneumococci, whereas IgA and IgE levels can be elevated. Now, IgM is also used in various lab diagnoses, where an immunofluorescence assay for IgM antibody is used, such as for the diagnosis of acute and congenital infections of Toxoplasma gondii. Toxoplasma gondii is actually a parasite acquired by the ingestion of cysts in uncooked meat or in food accidentally contaminated by cat feces. Congenital infections can result in abortion, stillbirth, or neonatal disease with encephalitis, chorioretinitis, and hepatosplenomegaly. Toxoplasma is one of the leading causes of blindness in children. IgM is used to diagnose congenital infections because IgG can be maternal in origin. Rubella virus is another disease in which IgM is used by observing the presence of IgM antibody in a single acute phase serum sample. This virus causes rubella and congenital rubella syndrome. Congenital rubella syndrome is characterized by congenital malformations. These malformations are widespread and involve primarily the heart, like patent ductus arteriosus, which is the abnormal persistence of an open lumen in the ductus arteriosus between the aorta and the pulmonary artery. After birth, the eyes, such as cataracts with cloudiness or opacity in the normally transparent crystalline lens of the eye, and also in the brain, like deafness and mental retardation. In a pregnant woman, the presence of IgM antibody indicates recent infection, whereas a greater titer of IgG antibody indicates immunity and consequent protection of the fetus.